All right, so last time we ended up talking about non-Mendelian inheritance, and the last thing we looked at was sex-linked or X-linked traits. Kind of ended with this picture of the human X chromosome. Today, there's just a few more non-Mendelian modes of inheritance we want to talk about, some more examples, and then we're going to look a little bit, the first part today, about how we can determine an inheritance pattern for non-Mendelian traits. So for Mendelian traits, since they're all dominant and recessive, simple Punnett squares work really well. But for a lot of these things that we've been talking about, Punnett squares don't work too great. Maybe for like incomplete dominance and sex-linked traits and things they do, but other things like pleiotropy and epistasis, that's got to be really difficult to figure out, like how is that inherited, from the outside if that you're given a specific trait and you're just making some observations. So we're going to look at pedigrees too today, because that is one way, especially like within human populations, we can get an understanding about how a certain trait of interest is inherited. But just first to finish up some of the non-Mendelian things we were talking about, there is also a phenomenon called X-inactivation. So in female mammals, they inherit two X chromosomes. And so one actually becomes inactivated during embryonic development, and it becomes inactive. It doesn't really do anything anymore, and we call it a bar body. Okay, that's why, both males and females are able to produce the same proteins and be healthy and have everything that they need, even though males only have one X chromosome and females have two, is that early in female development in the embryonic stages, one X becomes inactivated, and it condenses what we call a bar body. Um, what is really interesting is that which X chromosome becomes inactive is random in each cell that is alive at that point. So here's like an early embryo with 8 cells big, and maybe this is the point where X activation happens. And you can see it's kind of random amongst those 8 cells which X becomes inactive. So then when these cells multiply in mitosis to create a whole organism, there's kind of an assortment of cell lines that have come from these 8 original cells that have randomly X inactivation. So that's why, for example, you end up with uh, these cats, calico cats, that look like these have these tricolor patterns. They're all female because this color distribution is related to the X chromosome, and the patchwork nature is kind of like a roadmap showing you where the different descendants of these original cells that had random X inactivation ended up migrating to. So kind of if we think about it in this sense, the orange and the black coloration is a sex-linked traits in these cats. And when an X becomes inactive, it's no longer to express its allele, and so we get this piecemeal kind of different coloration on their fur. Male pattern baldness is what we call a sex-influenced trait. So, for example, it's an autosomal trait that's influenced by sex hormones as well as age. So, it's a dominant allele in males and a recessive allele in females, but it is even more so um, exemplified by the exposure to traditionally male sex hormones like androgens and testosterone. And so while females may inherit the alleles, it's expressed much in a much lower frequency in females because it's influenced by the exposure to its expression is influenced by exposure to male sex hormones. A lot of traits are controlled by not only genes but also the environment. Okay, so human skin color, for an example, is also influenced by both genetics and environmental conditions. So it's kind of like what we call a multifactorial trait. Um, Another example is in some flowers. So these flowers specifically, this difference in coloration is affected by the environment. The color shift in these flower petals is a result of the exposure of the flower to different pHs in the soil. So in a basic soil, the flower might have this deeper bluish color, and in a more acidic soil, it might have this more kind of reddish color, reddish pink. And so the environment directly can affect the expression of some traits. So there are environmental effects similar to the coloration in the fur of this animal here. And then second last thing is just talking about kind of what's called phenotypic plasticity. So phenotypic plasticity is the ability of certain organisms to express different phenotypes depending on the living and the non-living environment. And it involves kind of regulatory genes that switch on other genes and switch off other genes dependent on how the organism is stimulated. So as an example, many shade trees and shrubs produce shade leaves and sun leaves. So shade leaves are thicker and have greater surface area than sun leaves to help um, absorb more light. And it is the result of genes which determine the shape and the thickness of leaf uh, being switched on or switched off in response to the ratio of far red light to red light. So we learned that red is one of the colors that plants use very efficiently in photosynthesis. And so if there's a higher level of far red light than red light to a leaf surface, it's indicative of that leaf being more in the shade. And so that exposure to far red light will help it develop into a uh, uh, thinner leaf with a greater surface area. Or if it is exposed mostly to red light, it would have a thicker profile that would be not as great in surface area. And so that's an example of phenotypic plasticity, all right, changing um, in response to the environment. And then lastly, for non-Mendelian modes of inheritance, we have what's called mitochondrial heredity. So there are about 30,000 genes on, the 23 human chromosome pairs. That's about like 3.3 billion base pairs for every haploid cell. But there are about 27 genes that are located in the mitochondria. Because, like we said, mitochondria have their own DNA. 13 code for some proteins involved in cellular respiration. But the interesting thing is that 100% of all mitochondrial DNA is inherited from the mother. So all of your DNA that is found in all of the mitochondria in your cells is the exact copy of the DNA from your mother. You have none of your father's mitochondrial DNA. I think that's really interesting. Um, but so like when we talk about it, if we were looking for traits associated with mitochondrial heredity, we would only see them passed on from mother to offspring. Okay, so that's kind of it for talking about non-Mendelian inheritance. Now, one of the questions that always comes up is, okay, if these things are inherited in new and complex ways, how can we ever start to get a handle on what their a pattern might be? How can we predict, especially like in genetic counseling, how can we begin to predict probabilities of ourselves having these things or passing them on to offspring and things like that? And one really key way that we do that is through pedigree charts. Right, pedigree charts are used for a whole bunch of different things in genetics, and we'll talk about genetic disorders here at the beginning of the week next week. Um, but a pedigree chart is really useful, and we want to talk about just these three things briefly today about what it is and what they're used for. Their basic construction, so we know what the information they're representing, and how to tell the inheritance pattern of some simple Mendelianly inherited traits on a pedigree. So a pedigree is a chart of genetic history, kind of like a family tree over several generations. And scientists or genetic counselors can use it to find out about family history and analyze the chart so that they can make an educated prediction about how likely someone's offspring or a potential offspring would be to have a specific trait. So in pedigree charts, circles always represent females and squares represent males. And I have no idea how that got started, but these charts have been around for hundreds of years. Um, Connecting symbols together show some type of relationship. And so, for example, if two symbols are connected by a horizontal line, they mean that they are having offspring. And so, for example, here they have twins, and these are identical twins, so fraternal and maternal twins. So connecting a female and a male with a horizontal line like this shows that they are having offspring together. So in that case, the offspring are then drawn below that connection like this. So these two had offspring, and then the offspring, two females and one male, are shown like this. So these are brother and sister. These are siblings that we can trace back to the same parents, right? So here's an example of a pedigree chart that has 1, 2, 3 generations. So each generation is kind of like a new level or layer on this pedigree, this family tree. So these might be the grandparents, these might be the parents, and here's the children. Okay, and then you can see the aunts and uncles right here that uh, are the brothers and sisters of the two parents. When we create a pedigree, circles and squares that are filled in, like this one, represent individuals that have a specific trait, um, whether it is something bad like a disorder or just something that we're interested in. If and you'll see this very rarely. I'm not going to use pedigrees that have this. Sometimes if there's a dot in there, it'll tell you that it's X-linked. I'm just going to have you figure it out on your own using other evidence. And then sometimes, but not always, carriers will be half shaded in. Only half of it will be filled in. So we can use that information to determine if a pedigree chart is dominant and recessive and X-linked or autosomal. So the first thing that I would do is I would determine if a pedigree chart is autosomal or X-linked. If pretty much most of the majority of the people that are filled in that are affected, that have a specific trait that we're interested in, are males, that's a strong suggestion that it's going to be an X-linked trait. If it's closer to evenly distributed, that's going to be a signal that it's going to be an autosomal trait, so not on a sex chromosome. So as an example, this pedigree would most likely be an autosomal because we have 1, 2, 3 males, 1, 2, 3 females affected, kind of close to a 50-50 ratio. That's going to be an autosomally inherited trait. The second thing we want to do is determine whether the trait is dominant or recessive. If the trait is dominant, one of the parents of someone that's affected has to be affected as well, right? It can't skip over a generation, as we say, right? It's parent to offspring transmission, okay? However, if it's recessive, we can have an individual that has the specific trait that has parents that don't. That's not a requirement, but that's a strong sign that is recessive. It's what we call skipping a generation because those parents could be heterozygous. So for an example, this pedigree, it looks dominant to me. We've got direct parent to offspring transmission here, just like this. I would say this is dominant. This, on the other hand, look at this individual, this individual, this individual. They are all affected with whatever trait this is, but their parents, neither of them are. Neither mother or father in either sets of parents are affected. So this can't be dominant. This has to be recessive. Okay, so that's kind of all I wanted to talk about today. You'll have on your assignment some practice analyzing and interpreting pedigrees. And then when we get back together next week, we'll talk about genetic disorders and we'll do some other tests to help to determine heredity of certain traits.